Hello, my name is Lena Vincent. I'm a PhD candidate at the University of Wisconsin-Madison, and in this presentation I'm going to share some of the work our lab has been doing to try to understand how systems displaying life's key processes, like self-propagation and evolution, might have arisen in the absence of a prior living process. Our lab has been using theory and experiments to try to resolve this mystery, and to do this, we've developed a framework to try to explain how ecosystems of interacting chemical cycles could self-organize into systems capable of self-propagation and evolution, and how those systems might further complexify and eventually give rise to cellular life. And our hope is that this framework will yield insights into the origins of life from simple chemical precursors. Now, we believe that the ability to self-propagate and build complexity through adaptive evolution are two essential features of life. And to understand the origins of life, we have to try to explain if and how these processes can emerge independently of their specific biochemical constituents, including compartments, polymer-based genetics, and even specific metabolic pathways. Now, conventional approaches to studying the origins of life have historically focused on explaining the origins of these specific features, but as a whole, they fail to explain how those features could have arisen spontaneously in the absence of a prior living process, like evolution. As an alternative, the chemical ecology model, which is the one I'm going to present here, suggests that self-propagation and evolution could arise in chemical ecosystems independently of polymer-based genetics and even compartments. So what is this chemical ecology model? So to start, we define chemical ecosystems as sets of interacting autocatalytic chemical cycles. And an autocatalytic cycle is simply a chemical reaction or sequence of reactions where at least one chemical species is present both as a reactant and as a product, with more on the product side than the reactant side, and that's key. Now we call these member chemicals, denoted as M or mu in the diagrams I'm going to show you. And these cycles also require food, or F, which are present only on the reactant side, and generate waste, W, which are only present on the product side. Now our lab, in an effort led by postdoc Shen Peng, has simulated reaction cycles of varying complexity exposed to constant fluxes of food, and found that the behavior of even the simplest autocatalytic cycles can be approximated using logistic growth models, which basically just means that they behave much like populations of biological species. Even more interestingly, when there are pairs of interacting cycles that behave either as competitors, mutualists, or even predator and prey, the systems begin to display dynamic behaviors that resemble ecological interactions observed in biological populations, and these dynamics can be observed as changes in the concentration of member species over time. Now, a key consequence of this chemical ecology model is that it suggests that chemical ecosystems may be able to evolve as a result of combinations of different ecological interactions among many autocatalytic cycles, as well as random or stochastic processes like fluctuations of food species or other environmental factors. So we've also been working to explain how natural selection among chemical ecosystems can take place under plausible prebiotic scenarios, for example, in a seafloor or geothermal pool environment, recognizing that an important component of these settings are mineral surfaces, which have long been implicated in the emergence of life, as they do things like readily absorb or stick to organic compounds and have important catalytic properties. Chemical ecosystems stuck onto mineral surfaces, which we endearingly refer to as slimes or surface-limited molecular ecosystems, could differentially colonize surfaces and mutate as a result of rare side reactions, while the continual turnover of mineral surfaces could effectively select or enrich for slimes that are better able to colonize new surfaces. So this model shows us how, in principle, chemical ecosystems could complexify and evolve without implicating polymer-based genetics or even compartments. Now, an important part of our research has been to test this chemical ecology model empirically, and to do this, we've developed an experimental framework designed to generate and detect slimes under laboratory conditions, and we call this chemical ecosystem selection. The basic principle is really simple. It just involves combining a food-rich solution with mineral grains and deploying an analog of experimental evolution. And to do this, we incubate our ingredients to allow for putative slimes to establish themselves, and then we transfer a small subset of colonized grains, usually about 10%, to a new reaction vessel containing fresh food and fresh uncolonized mineral grains. And we repeat this over many generations to selectively enrich for variants, depicted in different colors here in this figure, that are better able to get from grain to grain and resist serial dilution out of existence. Now, importantly, we also routinely sample both the bulk solution and the mineral grains and analyze them to track any changes that might be occurring in response to the selection pressure. Now, the nice thing about this approach is that it's chemically agnostic in that it doesn't presuppose any specific chemistry and can therefore be used to test virtually any set of conditions. Instead, the general principle relies on the detection of changes in proxy traits across generations to infer the existence of slimes. And really, the only requirement is that the proxy trait be related either directly or indirectly to the rate of slime propagation. Now, response to selection here could manifest as a jump in the value of a proxy trait over one or several generations that is then sustained over further generations and can be seen here in this hypothetical figure representing the rate of propagation of a slime over time. Whereas progressive evolution would be demonstrated by gradual transitions to different proxy trait values. So now to give you specific examples of proxy traits we've been using to track slime formation in our lab and illustrate the chemical ecosystem selection procedure a little bit better, I'm going to show you some results of experiments we've conducted in our lab. So we've screened several sets of conditions, but for the purposes of this presentation, I'm going to focus on one specific recipe we used to validate the approach. And again, chemical ecosystem selection doesn't require any specific inputs, but here we chose ingredients that replicate what is known about the early Earth in some way. So we used a food-rich prebiotic soup modeled after spark discharge experiments, the recipe for which can be found in this table. But you'll also notice that we made additions of compounds like ATP as a chemical energy source, although we acknowledge that ATP is not a realistic prebiotic phosphate source. And for the mineral, we used pyrite, an iron sulfide mineral, which we ground up into powder and cleaned ourselves. And we carried out the experiment under anoxic or oxygen-free conditions, and we did so on 10 independent lineages that were exposed to 40 rounds of transfers, with each generation lasting about 2 to 3 days. And then we analyzed the replicate lineages periodically with controls that were strategically set up to only have a single round of transfer in their history to isolate the role of these transfers. One of the selected proxy traits we monitored was the concentration of free inorganic phosphate in the bulk solution at the end of each generation to estimate the amount of ATP hydrolysis that had taken place. And when we did this, we observed a distinct oscillatory pattern in the amount of free inorganic phosphate across generations. And there are several ways to interpret this pattern, the simplest being that a nonlinear or autocatalytic chemical system has arisen in response to the protocol. And interestingly, the theoretical work our group has done suggests that these oscillatory patterns might reflect the presence of predator-prey type dynamics in simulated chemical ecosystems, hinting at the possibility that interacting or coupled autocatalytic cycles might have arisen in our experiments. Now, on the mineral side, we also observed distinct fractal structures on the surfaces of pyrite grains from samples exposed to many rounds of selection, which are absent or rare in controls, meaning samples that only have one transfer in their history. And we did these observations by electron microscopy, and you can see some representative micrographs here for both experimental and control samples. And while we're still looking to determine the exact composition of these structures and their significance, and whether they're related somehow to the source of oscillatory patterns we saw in the bulk solution, this just further illustrates the fact that chemical ecosystem selection can be used to study complex spatial temporal dynamics that can arise in prebiotic mixtures. And while the results I've shown you are interesting and consistent with the appearance of interacting autocatalytic cycles, much more work is needed to determine whether they constitute true slimes by our definition. And for this, we would really need to demonstrate evolvability, and we plan on doing this by carrying out experiments for much longer, at least 100 generations, and performing more in-depth chemical characterization. And on the theory side, our group is working to analyze the effects of spatial structuring and heterogeneity to simulate the effects of mineral surface absorption on chemical ecosystem dynamics, which will enhance our ability to apply these models in the interpretation of results that we obtain in our chemical ecosystem selection experiments. So in conclusion, we hope that our chemical ecology model for the emergence of life will help advance our understanding of life as a general phenomenon and shed light on the origins of key life processes in relatively simple chemical systems. And I've shown you that our chemical ecology model can explain how chemical ecosystems of interacting autocatalytic cycles can yield complex dynamics and potentially evolve without the need for polymer-based genetics or compartments, and how chemical ecosystem selection might help us study the emergence of evolvable autocatalysis under laboratory conditions. And we've also already identified a set of conditions that yields promising results. And so ongoing and future work in our lab will be aimed at further understanding and expanding these theoretical and empirical models, and hopefully help the astrobiology community as a whole resolve the origins and fundamental nature of life. And with that, I'd just like to thank past and current members of David Baum's lab at the University of Wisconsin-Madison and Wisconsin Institute for Discovery, all of our collaborators at UW and beyond, and our funding sources. Thank you very much.